deuteromycetes, they include those fungi which reproduce only asexually. That is, they lack sexual reproduction. So they are known as a fungi imperfecta. There are some examples. First example is Coriototrichum. First example is Coriototrichum. It is a parasitic fungi which causes a disease called red rot of sugarcane. It is an important fungi. Coriototrichum is an important fungi which causes an important disease called red rot of sugarcane. Next example is Trichoderma. Trichoderma is also an important fungi which helps in biological control of other fungi. It helps in Trichoderma helps in biological control of other fungi. Next example is Fusarium. Next example is Fusarium. This is also very important as this is responsible for the discovery of first plant hormone. That is the discovery of first phytohormone which is derived from the fungi, that is gibberellin. Gibberellin is also discovered from the Fusarium as it reproduces asexually. It is an artificial group. Deuteromycetes is an artificial group which includes those fungi which reproduce asexually. That is, from which sexual reproduction has not been discovered. As the sexual reproduction discovered, they are placed in those classes, that is Ascomycetes and Basidiomycetes. Another important example is Helminthosporium. Another important example is Helminthosporium. It is very important and it causes brown leaf spot of rice. It causes brown leaf spot of rice, and this disease is responsible for Bengal famine. This disease is responsible for Bengal famine in 1945. So we have discussed the classification of fungi, that is, we divided true fungi into 4 types. That is, true fungi, they are known as Eumycota, and Eumycota, they are further divided into 4 types. First is Phycomycetes, which are commonly called lower fungi, and which mycelium is aseptate and cenocytate. Next is Ascomycetes, third is Basidiomycetes, and the fourth is Deuteromycetes. From them, Deuteromycetes, they are the most advanced fungi. Next, we discuss type of reproduction in these fungi. First is asexual reproduction. Asexual reproduction, first by forming different types of spores which are formed through mitosis. First is the zoospores. Zoospores, they are the motile and the flagellated spores. Zoospores, they are the motile and the flagellated spores. Example, they are generally found in moist fungi which grows in aquatic environment or in moist and the damp places like Saprolegina. Saprolegina is a aquatic fungi. Saprolegina is a aquatic fungi. Mostly fungi are terrestrial, but there are some fungi which are aquatic, and from them Saprolegina is an important example which reproduce asexually by forming flagellated spores called zoospores. Next method is sporangiospores. Sporangiospores, they are the non-motile spores found in terrestrial fungi. Sporangiospores, they are the spores which are formed in a special structure called sporangium. They are formed in a special structure called sporangium. And in sporangium, their spores are formed through mitosis, so they are called sporangiospores. And they are dispersed by bursting of the sporangium. And spores, they are liberated through wind. These spores are liberated by bursting of the sporangium, and the spores are liberated with the help of wind, and on reaching the suitable substratum they germinate. Example, rhizopus. Sporangiospores, they are the non-motile spores which are formed inside the sporangium. Third type is conidia. Conidia are also the non-motile spores, but the difference from sporangiospores is that sporangiospores, they are formed inside the body, whereas conidia are formed outside the body, that is exogenously formed. They are formed exogenously. Example, albugo. Example, albugo. As I already discussed that in albugo, at the time of reproduction, mycelium forms the finger-like projections called conidiophores. In albugo, at the time of asexual reproduction, mycelium forms the finger-like projections called conidiophores, and from the conidiophores, conidia are formed, and these conidia are formed in chains, and the arrangement of conidian chains is basipetal. That is, in albugo, conidia are formed in basipetal chains. That is, the arrangement of conidia is basipetal. Basipetal means that is youngest conidia at the base and the oldest at the top. That is youngest conidia at the base and the oldest conidia at the top. Next example is of penicillium. In penicillium also conidia are formed. In penicillium also conidia are formed, and on the branches from which conidia are formed, they are called stigmata. And this is the conidiophore, and the ultimate branch of conidiophore is stigmata, which bears chains of conidia. So conidia, they are present on conidiophores, whose ultimate branches they are known as stigmata. Next is sexual reproduction. As fungi, they are haploid. They are the lower organism, so their vegetative body is haploid. Fungi, they are haploid. So in fungi, the sex organs are known as antheridium. Antheridium is the male sex organ, and female sex organ is oogonium. So sex organs and during the sexual reproduction, two sex organs are known as antheridium and oogonium, in which male and the female gametes are formed. So male gametes are formed inside the antheridium, and the female gamete is formed inside the oogonium. There are three important steps in the sexual reproduction. Three important steps. Three important steps. First is plasmogamy. Gamy means fusion, and plasma means fusion of cytoplasm of two different gametes. That is fusion of cytoplasm of male and female gametes. First step is fusion of cytoplasm. Next important step is karyogamy. Karyo means nucleus, gamy means fusion. Firstly, two gametes, cytoplasm fuse, and after the fusion of cytoplasm, two nucleus also comes together and they also fuse. So this process is known as karyogamy. That is fusion of two nucleus. That is karyogamy means fusion of two nucleus, that is of male and the female gametes nucleus. As fungi are haploid, so after fertilization, it results in the formation of a diploid zygote, and this diploid zygote give rise to the diploid body. So for forming haploid body, there is meiosis. So third step is meiosis. Third step is meiosis, which results in haploid body, which results in the formation of haploid body. These are the three important steps in sexual reproduction. That is, first is plasmogamy, which means fusion of cytoplasm of male and the female gametes. Second step is karyogamy, that means after fusion of cytoplasm, two nucleus, that is of male and the female gametes, they come closer and there is a fusion of those nucleus, and this step is known as a karyogamy. And the third step is meiosis. Meiosis, due to karyogamy, it results in the formation of diploid cell, and this diploid cell, after reduction, that is after meiosis, it results in the haploid body, which forms the vegetative body of the fungi. Three methods. From the plasmogamy, after plasmogamy, it results in the formation of, that is, a cell with two nucleus. It results in the formation of two nucleus. And by karyogamy, it results in the formation of a diploid nucleus. So there is no time gap between, generally not time gap between plasmogamy and karyogamy. That is, plasmogamy is immediately followed by karyogamy. But there is some exceptional case, that is in basidiomycetes, and I have already discussed. In basidiomycetes, which includes Puccinia and Agaricus. I have already discussed basidiomycetes, which includes Puccinia and Agaricus. It is an exceptional case in which there is a time gap between plasmogamy and karyogamy. So for a time period, it results in the formation of a cell with two nucleus, that is monokaryotic. A cell with a single nucleus is called monokaryotic. When these two cells fuses, it results in the formation of a two-nucleated cell, and this two-nucleated cell is called a dikaryotic cell. So in them, this dikaryotic forms a different type of mycelium which is found in Agaricus, known as a dikaryotic mycelium, which is found in Puccinia and Agaricus only. Generally, mycelium is monokaryotic. Different methods of sexual reproduction. First is gametangial contact. Gametangial contact. Gametangium is the general term of two sex organs. Gametangium is a general term given to the male and the female sex organs. So, at the time of sexual reproduction by gametangial contact, two sex organs, that is male and the female gametangium. This is the antheridium. This is the antheridium, and this is the oogonium. They comes in close contact, and this antheridium forms a tube-like structure which enters the oogonium, which is known as a fertilization tube. This forms a fertilization tube, and through this tube the male nuclei, antheridium contains the male nuclei and the oogonium contains the female nuclei. So through this fertilization tube, male nuclei enters the oogonium, and on reaching the female nuclei, this tube bursts, releasing the male and female nuclei, resulting in the formation of diploid zygote. And this diploid zygote is known as a oospore. After fertilization, a diploid structure called zygote is formed, which is known as a oospore. Example through which gametangial contact occurs is found in albugo. Example is albugo. In albugo, sexual reproduction takes place by gametangial contact, in which male and the female gametangium, they makes a contact, and after fertilization, this fertilization tube disappears and the antheridium separates from the oogonium. That is, antheridium and oogonium, they don't lose their identity at the time of reproduction. This type of reproduction is found in albugo. Next type of reproduction is gametangial copulation. This is found in Rhizopus. In this, the two gametangium, that is male and the female gametangia, these are the male and the female gametangia, which are also denoted by plus and the minus type, that is, they are of two different strains. So in them, at the time of reproduction, they forms a projections, both the gametangia, they forms a projections towards each other, and in which more of the nuclei, they are present at the terminal end, known as progametangia. And after coming in close contact, the cross walls between the male and the female, this disappears and their content get intermingled. That is, at the time of sexual reproduction, both the gametangium, that is male and the female gametangium, they lose their identity and they maintain a common contact, and it results in the formation of multinucleated structure after fusion known as a zygospore. So in Rhizopus, after fertilization, a sexual spore called zygospore is formed. So by gametangial copulation, there is fusion of two gametangium which loses their identity, whereas in gametangial contact, the two gametangia, that is male and the female gametangium, they don't lose their identity, whereas in gametangial copulation, male and the female gametangium, they loses their identity and it results in the formation of a zygospore. Third method of sexual reproduction is spermatization. Spermatization is a method which takes place in Puccinia. In them, at the time of sexual reproduction, one of the hyphae get specialized to form structure called male gametangium, that is a distinct male reproductive structure is not formed. Only at the tip of one hyphae, a spore-like structure called spermatia is formed, and this spermatia, it acts as a male gamete. Spermatia is a male gamete which is formed on the one hyphae, and from other hyphae is formed a female structure called trichogyne. And in the trichogyne there is a egg. In the trichogyne there is a egg, and this spermatia, they are carried through wind, and this enters the trichogyne and this fuses with the egg and results in the formation of a diploid structure. This is the spermatization which occurs in Puccinia. In spermatization, a spore-like structure called spermatia is formed, which is known as a male gamete, and this gets dispersed with the help of wind and it enters the trichogyne, which acts as a female reproductive structure which contains an egg, and after fertilization it results in the formation of diploid zygote. Next method is somatogamy. Somatogamy is a method in which there is no distinct sex organs are formed. That is, sex organs are absent and sexual reproduction is accomplished by fusion of two hyphae of opposite strains. That is, somatogamy means fusion of two somatic cells. That is, this is the hyphae of one type, that is of one strain, we call it plus, and this is of minus. At the time of somatogamy, there is a fusion of two cells. It results in the fusion of two cells and it results in a dikaryotic cell, and it results in the dikaryotic cell which contains the nuclei of two opposite types. That is, a nuclei of two opposite types. Somatogamy is a method in which there is no distinct sex organs are formed and sexual reproduction is accomplished by fusion of two hyphae which are of two different strains, and such results in the formation of a dikaryotic cell. This occurs in Puxi, agaricus. It results in agaricus and Puxinia. As a result of sexual reproduction, it results in the formation of different type of spores, sexual spores. That is, first is oospore. Oospore, it is a diploid which is found in albugo. Second is ascospore. Ascospore is haploid which is found in yeast. Third type of spores. These spores are formed as a result of meiosis. Zygospore is also a diploid which is found in rhizopus. And fourth is basidiospore, which are also haploid and they are found in Puxinia and agaricus. So, as a result of sexual reproduction, there are four types of spores are formed. First is oospore, which is a diploid. Ascospore is haploid, which is formed as a result of meiosis. Whereas zygospore is a diploid and basidiospore is also a haploid, which also results due to meiosis.